All right, so this is midterm review, day 3 of 4, and tomorrow will be the last session. Tomorrow we'll be focused on all Unit 4 stuff, so stuff that would have been on a Unit 4 test, but, you know, we're doing a midterm instead. So a lot of your questions will be pulled from Unit 4, so I'm sure a lot of you will be paying very close attention tomorrow. Moving on. So first things first, we want to talk about cell respiration. I'm going to try to slow down when I write today because I don't feel like the clock's like trying to eat me today. So cell respiration. This will be chapter 9. 9 or 10. I'm not sure. So the formula, by golly, I hope you remember it. Please tell me you do. I hope so. Okay. C6H12O6. That's sugar. It's a generic form of sugar. Keep in mind, real cell respiration probably doesn't actually use glucose. It probably has to break it down from starch because most plants store their sugars in the form of starch. But whatever. And that's going to be 6O2. And that is going to make 6 CO2 and 6 awatas. Right? And this is the balanced equation, so try to keep in mind for every one glucose molecule that we have here, there are 6 oxygen molecules present as well. And this would be a balanced equation. You're not going to see any questions on that. But basically, I guess you should... Now we'll just keep going. So this whole thing also makes energy. The whole purpose of this is to make ATP. And it makes a good deal of it. It's somewhere between 34 and 36 ATP. Does it make other forms of energy? Yeah, it does. But the main output that we use, the main currency that we use in our body for energy, it would definitely be ATP. This is also really, really, really important because the process of cell respiration happens in a few key stages. Does anybody here remember what has to happen before? Oh, I should mention this. This is actually aerobic respiration, not just respiration, but aerobic respiration. Does anybody remember what happens before aerobic cell respiration? Has something to do with this stuff right here. No. Though photosynthesis is related to this. This is too complex. Our body can't process this. So what we need to do is we need to break this down during a process called, just trying to give it to you on a little plate here. It sure is, yeah. It's glycolysis. Glycolysis is are the two root or origin words there. Glyco meaning sugar and lysis meaning breaking. So we're actually breaking that sugar down to make it more digestible. What do we turn that sugar into after this process? What's our 2 end products? I've given you a hint. It's 2. Uh-huh. No. What's that? 2-pyruvate. Okay. And there's also a little bit of something else made. It is. It's 2 ATP, as a matter of fact. So it makes just a touch of cellular energy. Those pyruvates are really the things that feed into this formula. Those are really those things. But anyway, they get involved in the actual process of cell respiration later on. Now, is glycolysis, is this aerobic or anaerobic? It is anaerobic. And that means it takes place where? Correct. It does not take place in the mitochondria. It happens in the cytoplasm. Okay, that's a key thing. It happens outside of the mitochondria. As much as you may want to think all forms of cell respiration take place there, that is not true. So glycolysis will eventually feed into a process that is in the mitochondria. I don't want to draw it, so I'll just do this. This will eventually lead to some things happening, and this will feed into the first stage inside the mitochondria. What's that called? It's got two names.
Krebs cycle / cycle.
You're right. It's the Krebs cycle. Or you can also call it the citric acid cycle. Okay. Does this make a lot of ATP or not so much ATP? Yeah, the pyruvates will then get fed into this and be part of the citric acid cycle. It only makes a small amount of ATP. Generally speaking, that's about 2. There are also some other things involved too. Do you guys remember any of those other fun molecules that are involved? You guys are crushing it right now. That's good. Glad we're doing this review session then. It also produces... What? NADH, and then also, we'll say this way. So these things are produced here as well. So we have some ATP, but we also produce NAD and FAD, or FADH2 and NADH. So those things will be produced in the Krebs cycle. What do we refer to these as? What do they do with electrons? Yeah, they're electron shuttles. So they carry electrons around for us because we want to transfer some energy for us. I should also go ahead and mention, I'm not going to break down the citric acid cycle like what we did previously. You don't need to know every single step in there. But it's called the citric acid cycle because there's something involved called citrate. And then citric acid will basically be converted. Every once in a while, we'll break this down and we'll have a little byproduct. But what is also produced at the end? What kind of gas? Carbon dioxide. There it is. So we start breaking down sugars because it's carbon and oxygen and hydrogen. C and O form CO2. So we can form carbon dioxide gas at the end of the Krebs cycle. You doing okay? Can we move on to the electron transport chain? You sure? Well, by that I mean I'm going to click this and the screen will clear out. Maybe I should read a little bit more.
Stop. That's not reading.
Sorry, I had to wet my whistle before I read.
Ew. You better whistle the rest of the class. But you can only speak and whistle, so think of that. Morse code. Yes.
All right. So we got the electron transport chain. Well, whatever. So basically, let me give you the long and short of it, actually. Let's say... I don't want to waste too much of your time, but... All right. So, in this process, we're going to move hydrogen ions through the mitochondrial membrane. If I were to look at it, it looks kind of like an outcropping there. And remember, there's like these are cristae, so there's all kinds of little folds like this on the inside. That's why it's a double membrane organelle. And our goal is to pass electrons through this and eventually pass them out of this. This movement is really important because we want to move these hydrogen ions. And they're going to move against the concentration gradient. So there's already a lot of a really high concentration gradient in here. The other thing is we do get some byproducts here. We start to see, we form ADP as the opposite of what ATP is, because now we're using up that energy and we're sending it back. We also are sending back well, anyway, we're sending back FAD.
That means you're making it?
Oh, it's just like the inverse of FAD H2. So
No, but like you're making
yeah, these are the things I guess that are being produced, which is just because we're using up whatever was there.
Okay.
So yeah, we're I guess creating these. Yes, ma'am.
Don't you start with FADH2?
FADH2 is what you start off with, and that's what we did in the last step, the Krebs
so
cycle.
Do we get rid of the two of them or just the one?
Yeah, just this.
Okay.
And then we'd be sending back, I believe it's NAD plus. Anyway, so those things are going to be created, and they're just like the leftovers. They're going to be sent back to the beginning and repeated. We also get a byproduct of water. Water is being produced through cellular respiration. That's what's happening here. Once we move against the concentration gradient, we create an imbalance, and we basically call this chemiosmosis, which is this movement here. But also, we have something called phosphorylation.
So can you like define chemiosmosis?
Yeah. Ah. I'm just going to write CG for concentration gradient. Do you follow?
Okay.
So the hydrogen ions are moving against the concentration gradient, and I've kind of pointed to it right here. So there. Anyway, I think it's phosphorylation like that. Anyway, oxidative phosphorylation. Oxidative phosphorylation is basically when we add I see a confusion there. I'll clarify in a second. When you add inorganic phosphates to ADP for converting to ATP. All right. So I think my confusion up here, I made a boo-boo. I'm going to fix this. You're going to get all mad at me. I wrote ADP instead of ATP for some reason. And this generally ranges from about 30. It can be a slightly variable level. It could be 32. There's not really a precise number because the number of cycles that turn changes the numbers of ATP that are produced in the end. But yeah, there you go. So we're making a whole bunch of ATP in this process. Yeah.
PI.
Inorganic phosphate. So it's a P for phosphate. The I stands for inorganic. It just means it's not attached to a carbon. It should be a free-floating phosphate too. Doing okay? Moving on.
No.
I'm reading. Oh, these words are good. Really good words on this paper. Good stuff to read. I started reading a book by Ernest Cline called Armada.
Oh, really?
Yeah,
How is it?
it's starting off okay. If you've read Ready Player One,
Yeah,
it's got the same feel.
it does.
Hmm
Are you
It's all
gonna
about
see the
video
movie when it
games
comes
now.
out?
What?
Are you gonna see the movie when it comes out? Ready Player One? March?
My mindset is I know it's not supposed to be anything like
Yeah.
the book,
'cause that's what Casey
so
missed out
just
on.
I'm gonna go and watch it and be like, cool. I'm just on it for the ride. Let's
Yeah.
see where this goes.
Because like the ads are like that didn't happen. What's he
Yeah.
doing?
Why is there a giant race? What's happening?
But it still is cool.
Yeah, it does.
What's the I again? I know you.
Or inorganic phosphates.
Okay.
So the I is inorganic, P is for phosphate. Yep. And if you read Armada, a lot of you will really like it because it talks about a boy who's a high school senior, and he looks out his classroom window and something remarkable happens.
Your Armada?
Yeah, the book Armada.
Oh, okay. I read at the end of Ready Player One, he had like the first chapter of that book in it, and I was like, oh, that sounds cool, but then I totally forgot the name, and so you're like, oh, I looked out his window, and I was like, remember the kids tripping out.
There you go. All right. Can I move on now? Because I'm done reading. Photosynthesis. I believe it is. I think we went in order, though you may want to double check that when you're at home. So photosynthesis. You should know the formula for photosynthesis. It's the same thing as cell respiration, but backwards. Do I have to write it? No. Okay, good. That's all I need. So their whole thing is that they're capturing solar energy to convert it to chemical energy. So this conversion is all photosynthesis is. We're trying to capture that energy from the sun and store it in a chemical way. That chemical is sugar. So this process, wildly important, is going to create good amounts of sugar, which of course, if you look at the formula there, you will get it. I should also, I should have done that previously. We'll go back. But anyway, we have two halves to this thing. We have light-dependent reactions. And we have light independent. Okay? Light dependent is kind of the reverse order of what we saw in cell respiration. So instead of ending with the electron transport chain, we're kind of starting with it. Okay? So this is when we'll just say light energy excites these things called chlorophyll and helps shuttle electrons through the thylakoid membrane. Hey, this is a membrane too. Just like the electron transport chain used the mitochondrial membrane, now we're using the thylakoid membrane. Try to keep in mind the thylakoids are the individual pancakes, right? They are stacked up with other pancakes, but each individual pancake, that is going to be a thylakoid. We're talking about that membrane. That's what's going on there. So that's light-dependent reactions. They're going to be moving through there. When we think about the byproducts from light-dependent reactions, because we're feeding in CO2, we're going to break this down and have O2 as a byproduct. So input, output. Okay? We'll shuttle some electrons, we'll transfer some energy, but that's all going to be sent over to the light-independent reactions. And the light-independent reactions, also known as the Calvin cycle. Bless you. This is going to take in... Oh, crap. I'm so sorry. That's water. I'm a goose. Our input here is going to be CO2. And our output will be sugar, or C6H12O6. So this is going to be the attachment of carbon to, I guess we'll just call them intermediate molecules. Only found in the Calvin cycle. Okay? So you want to, as best you can, just try to remember those inputs and outputs. You might see a question that pertains to that. Okay? Yeah. We also talked about how there's different kinds of chlorophyll. And we mentioned some chlorophyll absorbs certain wavelengths better than other ones. Don't just think there's one type of chlorophyll. There are multiple kinds. Doing good? I'm going to read. Just reading. Here I am, just a reading fool. Good times. Just reading. Just reading ahead. Reading a little bit more. A few more words left on the page. Just reading them out. Oh, they say words and science. Mostly.
Word science.
Words of science.
You said you had 2 words in science.
I said a few. I said a few. I said a few. Oh, look at me being all lucky. All right, so then we have anaerobic respiration. I guess this would be, yeah, still the same unit. I can't remember what chapter it is. I want to guess it's part of chapter 9. I believe cell respiration and anaerobic and aerobic are in the same chapter, I believe. Okay, so we already talked about glycolysis, right? Glycolysis can be kind of repurposed in 2 ways. We have alcohol fermentation. And we have lactic acid fermentation. I'm just going to be lazy. Okay? Glycolysis, if we forced it to continue, it would be a form of fermentation. So a question you may want to reference, or a point of reference, would be if somebody were to ask you, is fermentation making energy? Technically, no. It's actually forcing glycolysis to continue. It just creates different end products. The end product in alcohol fermentation, of course, is going to be alcohol, but also CO2 gas. All right. Which, of course, gets used for like a whole lot of purposes in like commercial or industrial baking and stuff like that. Even for creating just like alcohols that are consumed as beverages. And then we have lactic acid fermentation. It makes this thing called lactate, which is basically just lactic acid. It just has another name. And this causes the burning sensation in your muscles. And for alcohol fermentation, we'll say baking or alcohol made. So glycolysis, if it's forced to continue, can lead to these. Remember, these are done whenever an organism doesn't have enough O2. Some organisms depend on this, but some organisms can kind of turn this off and on. Generally speaking, we'll just say if O2 is brought into this environment, if oxygen is brought into this environment, this process will stop. So you guys might know of like alcohols, like let's say a wine or something is being made, they should be sealed. And that's because it stops oxygen from getting in there. It's not like they pump the thing out beforehand, but so much CO2 is produced that maybe the container might break. If that happens, you end up getting something closer to a balsamic vinegar instead of actually making a wine. So controlling the amount of oxygen that are mixed in with these organisms is a really big deal. That's all you would really have to know for anaerobic respiration. Doing okay? I need to read? Okay. Mm-hmm. Mm-hmm. I should just write this off to the side. It does use another energy molecule. You might see a question that asks you about what kind of energy molecule is used in forms of fermentation. NAD would be it. It recycles that, so it keeps sending it back to the beginning. And it only produces a little bit of ATP. It produces the same amount of ATP as glycolysis. So it doesn't make a lot, but it's enough to keep a small organism going. For us, whenever we're oxygen deprived, or I should really phrase it as just having an oxygen deficit, if you're doing a physical or strenuous exercise and you're using muscles, your body can't take in enough oxygen to make it work. So lactic acid is going to build up because your body's forcing glycolysis to continue. Doing okay? Still reading? No more words on my paper? No? I'm reading. Yes, I am. Don't judge me. I'm reading the newspaper. See? Okay. Let's go ahead and hit up some easier, shorter, easy to digest topics. Let's start with this one. And by the way, we're moving on to Unit 4, if you couldn't tell. So, yeah, good times. So, with the cell cycle, where's my... Oh, you jerk. Hey, it wasn't super bad. There wasn't a whole lot of stuff on there.
Yeah, what if it was an entire page?
I just quit. I just quit. That would be the end.
Please don't.
We're already ahead of pace, so you know it's all bonus points at this rate. Okay, so the cell cycle. I don't know. We'll just do it this way. Hopefully, this will be. Gosh, I'm really trying. All right, there we go. So we're gonna start off with one cell. We'll end up with two cells. A good chunk of this cell's life is going to be spent in gap 1, synthesis, and then gap 2. Remember, at the end of gap 1, I'm only drawing this to do this stuff here. Yeah, there's 3 checkpoints that occur. Okay, oh, there's a way that you could kind of repeat this cycle. That's gap 0. We'll talk about it in a second. Anyway, these are checkpoints. These checkpoints check for mistakes in replication. Okay? Not just replication, they're just trying to prepare itself for replication to make sure everything's the way it should be. In the cell cycle, I tried to draw them roughly about this size, but you'll see a huge chunk of their time is spent in gap 1 and gap 2. A little bit of time spent in synthesis because this is actually DNA replication. We're actually replicating the DNA, and it's got to go through a checkpoint before it gets to that area. Then it goes through gap 2. Before it even makes it to mitosis, it does a final checkpoint and says, am I good? Do I have any irregularities that don't need to be copied? If it's good, it moves on to mitosis, M phase. And in the middle of that, right before anaphase, which I'll write those down for you in a second, right before anaphase, it makes a final check to make sure the chromosomes are where they should be. This prevents some chromosomal abnormalities, but of course they can still result. Anyway, then it'll finally split apart. So those would be the 3 checkpoints, 1, 2, and 3. You guys remember the stages of the entire cell cycle. It's going to start with interphase. And this is when the cell will perform its normal functions. It'll do normal cell things. It'll grow. That's why we have G1, S, and G2 in that order. But then after that, we go to mitosis. And mitosis is going to include prophase, metaphase, anaphase, telophase, and cytokinesis. Try to keep in mind, technically, cytokinesis is its own separate thing. It's really not part of mitosis. It would be like at the very tail end here. But anyway, prophase, you should have a basic understanding what's happening there, right? What happens? Good. Good. Prophase, yeah.
Isn't that where it starts to duplicate DNA?
Nope, that would happen back here in synthesis.
Oh. Doesn't the nuclear envelope disappear?
Yeah, so the nucleus starts to disappear. I'm just going to list it over here. Nucleus dissolves, right? And also the chromosomes will condense. Now, back in honors, this is about the limit of what you had to know, but you should be aware that the spindle fibers, or those, they're basically modified microtubules. Those things, the spider legs, those are going to start growing at this point, and they're going to start moving to those poles of the cell, the north and south poles of the cell, and that's really important too. So spindle fibers form, or mitotic spindles if you want to be correct, but spindle fibers form. Okay. After that, we end up with something in metaphase that forms the metaphase plate. Right? We could even actually say there is a secondary stage in between these. I'm just going to write this here. It would be prometaphase. It's just like what happens in between prophase and metaphase. And the chromosomes are just moving. They're getting closer towards the middle, but they haven't formed the metaphase plate yet. But the metaphase is known for having that metaphase plate, which means those chromosomes would line up in the middle like that, right? A straight line. The mitotic spindles are going to start to grow and stretch across there. Then we get to anaphase. Anaphase. What happens?
Tear the children apart.
Yes, so the technical phrasing is chromatids. Notice I'm not saying chromosomes, I'm saying chromatids separate. Okay? Because remember, half of a chromosome is a chromatid. To describe that for you again. Okay? This whole thing is a chromosome. But just this half, oh boy, I tried. That's a chromatid. So once those halves separate, we say we've got 2 separate chromatids. Does that get confusing later on with chromosome number? Yeah, it does, especially with meiosis, because you can have one chromatid that we now start calling a chromosome again, but the real secret is just counting the number of centromeres. So remember this middle region that's found right there. Let's try that again. Hopefully it looks 3D. This is the centromere. It's merely the center of the chromosome. Yeah? Okay. Ringing bells? Feeling good? Okay. So once you go through anaphase, then you can hit up a wonderful little telophase. And this is when the cytoplasm starts to divide. There's like this little contractile ring that forms, and this would be the cleavage furrow. That is what it's called. But there are different versions. I'm just going to make a little note off to the side here. In animals and plants, they're called different things. Do you guys remember what it's? Well, I just kind of gave it to you. Cleavage furrow. In animals, but what do we have happen in plants? Say again? Yeah, the cell plate. This is the formation of the cell wall between the two new cells. Cleavage furrow, it just like splits the membrane, so they'll just have two separate membranes. They never had a wall in the first place. So that would be telophase. Cytokinesis, of course, is just going to be the cutting of the cell cytoplasm. And then you're good. You have two separate cells. So the stages that we just mentioned here, mitosis, that's involved in some organisms, right? Not all organisms reproduce this way, which leads me to comparing this to another form of stuff. But do I need to read? I need to read a little bit. I should read. Yep, this paper's so good. And my cup is so dry.
Someone's not staying hydrated.
Oh, I've done great today.
Yeah?
Great. And yesterday, when I got home, all I did was drink water. All I did. I remember laying down on my phone. Like, I didn't lay on my phone. I laid down with my phone, and I remember snoring, and I woke myself up, and I was like, no, you have things to do. And I went and I did all the things I had to do. And then I went to bed around 9 p.m. last night. Yeah, I woke up at 1:30, and then I promptly went back to sleep and woke up at like 6:20. Yeah, I did. You're right, Opal. I did. Yeah, I kind of did. Yeah, I kind of did. I'm here. I wasn't late. Still getting paid for today. All right, so moving on. Did I read enough? Yeah? Okay. So we mentioned this. Different types of organisms use different types of things. Mitosis, it really works well for diploid organisms because it forms what? When you go through mitosis, you start off with a diploid cell. What do you form? They're identical, which means you form now two diploid cells. Okay? Another process would make haploid cells. In another one, and by the way, this is found in eukaryotes. In prokaryotes, we use this process called binary fission. Okay? One of the big reasons why binary fission is used by prokaryotes is because it doesn't require all the individualized steps that we find in mitosis, because prokaryotes are really simple organisms. They just have a small circular strip of DNA that kind of connects to itself. We have separate chromosomes that are all complicated and messy, so we'd want to pull apart each of those chromosomes in the proper way. Bacteria don't have that, so they just copy that ring of DNA, and they use, basically, when we talk about replication, we'll do this. You'll see that they produce their own little threads of DNA from this little molecule that kind of runs along that little ring, and it makes a copy, and it's floating, it's free floating. They don't even have a nucleus, right? Yeah.
So like when you look at the cells like splitting like under a microscope
Yeah.
for like our
our cells,
Yeah.
you can see like the chromosomes splitting. Like, how does it look with them if it's just reflected?
Honestly, if I were to draw it, it would look like, well, their DNA looks probably like that. Like it's a circle because it's connected, but it's like all twisted up. It's like whatever.
Yeah.
If you were to see this happen, you would see like, you just see these things separate from each other, and then you'd see just two new cells form.
Okay.
Like, it's dumb. There's no like pretty stuff. There's no arrangement. It's not precise. Things can even get screwed up in that process a little bit easier than it can in mitosis. They don't have the same regulatory checkpoints that we do, which actually explains why we see mutation rates. One of the reasons why we see mutation rates so high in bacteria, because, well, they don't have a whole lot to change, so one little tweak makes a change.
Yeah.
But also, they don't have the same regulatory measures that we do.
Okay.
Yeah. Was there another question that I missed? Thought I saw another hand. Okay. So binary fission is definitely used in prokaryotes. It's still, well, I don't even want to say that. Well, yeah, I do. It still make diploid cells.
Can you explain the difference between
Diploid and, yeah.
Does it make two diploid cells?
Yeah, each cell would be a diploid cell. Sorry. Haploid. So this means you have two copies of basically your DNA, your genetic info. This only has one copy of your DNA. That's the difference. So when we talk about a chromosome, that's one and that's one. It's the worst line ever. But yeah, that's one copy. That's the other copy. So that's why when we split it in mitosis and we've got like lots of these things, each side is still getting a full set of DNA. Nothing's being shortchanged. So diploid definitely have two copies that are stuck to each other. Word? All right.